think it's time to start. So good morning or good afternoon or good evening, everyone. Thank you for joining us for this third webinar offered by the Groupe Polyphénols. Polyphenols. The Groupe Polyphénols started to develop this series of webinars in Polyphenols Research last year, and with presentation about polyphenols in soil systems and characterization of polyphenols. If you did not have the chance to participate in the first two webinars, you can still have access to those recordings on the website of the Groupe Polyphénols. I'm Dr. Aude Watrelot. I'm an assistant professor and extension specialist of enology at Iowa State University in the United States. I'm focusing my work on polyphenols, especially in red wine and how it's related to the quality of red wine. I'm a board member of the Groupe Polyphenols, and I'm the host of the webinar today. So today the topic is about polyphenols and food and beverage quality. And I'm pleased to welcome our two fantastic speakers today, Dr. Catherine Renard and Dr. Elsa Brandão. They will both present on their research topic that is related to the impact of polyphenols throughout processing, food processing, and the quality of food and beverages. So we'll start the webinar by a presentation from Dr. Catherine Renard on polyphenol from fruit and fruit to juice with a specific focus on apple, pear for about 25 minutes. Then we will continue with the presentation of Dr. Elsa Brandão on polyphenols and quality and mouthfeel properties of wine for about 25 minutes. This will be followed by about 30 minutes of questions and answers. So if you have any questions during those presentations, please type them into the chat box, and we will answer them at the end of both presentations. So today, that's my great pleasure to introduce Dr. Catherine Renard, who was my former PhD supervisor at INRAE Avignon a few years ago. So Catherine Renard obtained her PhD in 1989 after a master in physicochemistry of biomacromolecules at University of Nantes and initial training as a pharmacist. Since 1990, she has worked as a scientist at INRAE, first in Nantes on physicochemistry of plant cell walls and pectin, then in Rennes on cider processing and polyphenols, and then in Avignon on processing and quality of fruit and vegetables. From 2011 to 2018, she was head of the JRU Safety and Quality of Products from Plant Origin. She coordinated FP7 project OptiFel, which is Optimized Food Products for the Elderly, and some French national project. Since 2018, Deputy Head of Transform Division of INRAE, Science and Processing of Food, Bioproducts and Waste, Head of Carnot Qualiment. Her area of expertise is microconstituents of fruit and vegetables and their fate during processing, notably characterization of cell wall polysaccharides, more specially pectins, plant polyphenols, notably procyanidins or tannin, interaction between polyphenols and cell walls, impact of process on phytomicronutrients and vitamins C and folate, analytics and kinetics. She is author of more than 200 original publications in peer-reviewed journals, mainly food chemistry and applied chemistry. She has been the organizer of the European Workshop on Apple Processing in Rennes in March 2005, and of the first and second Euro-Mediterranean Symposium on Fruit and Vegetable Processing in Avignon in 2011 and 2016. So now I will make you presenter, Catherine, and you will be able to share your screen.
Thank you, Aude, for this very complete presentation. And I'm going to start. I hope it works. Yes, looks like it.
It works well.
With my presentation on polyphenol from fruit to juice, how variability is modulated by processing. This is work which has been carried out in INRAE, in Rennes, and in Avignon. And thank you for, and I will start. I will give you first a few words about the origins of the variability of polyphenols in fruits the genetics, and also how the polyphenol change during maturity and growth and storage. And then I will talk about what is really the topic here, which is the processing and how this will act in transfer of polyphenol from the fruit to the food, either a juice or a cider, because I'm going to talk about apples, as I will talk about wine later, and also a few words when you are not pressing, but using only a heat processing. I'm going to talk mostly about 6 types of polyphenols, some hydroxycinnamic acids, mostly chlorogenic acid, and a lot of flavonoids, dihydrochalcones, which are quite typical for apple, flavonol, anthocyanins, and also quite a lot on flavanol, be it the monomeric flavanols, the catechins, or the oligo and polymeric flavanols, which are procyanidins. As you can see here, they each are in a colored background, and I will use the same color convention throughout the speech. So if you see something red, it's a procyanidin. If you see something orange, it's a flavonol, etc. So I will start with genetic differences. There's major qualitative differences between species. And you have here 4 examples of fruits, apple, apricot, plum, and strawberry. And as you can see, some of the polyphenols are specific, like, for example, dihydrochalcones in apple, so there's some minor amounts in strawberry, and some are very common, like, for example, the hydroxycinnamic acids, the flavonols, most of the flavanol, the anthocyanins in the red fruit. So they are present in many different fruits. The equilibrium between the different classes vary from fruit to fruit, and this will give a typical signature each time. You have differences between the different species, but that's not the only differences which you have. And for example, if you're talking about here, it's pears. As you can see, there are 2 major differences when you compare the pear flesh and the pear skin. There are qualitative differences, but there are also very large quantitative differences, as the scale is 5 times larger for the skin than for the flesh, which means it's much more concentrated. Polyphenols are much more concentrated in the skin, which could be expected, because they are here also for protection of the fruit. And when you look here, that's pear. I like pear because it's a very simple polyphenol composition. It's almost only procyanidins and hydroxycinnamic acid. There's some arbutin, but it's a very low amount. And if you look at the skin, then you will see 2 more classes appear, which are the monomeric catechins, very low amounts, but they are present here, and the flavonols here. What I wanted to show also on this slide is that you have an incredible difference in quantitative content between the different varieties of pear. Here you have the example above of French table pears, and some of them have very low polyphenol content. Some of them will have more. You also have here an example of a series of Tunisian table pears, and you can see that they have already quantitatively higher amounts on average, but still a very large variety. I mean, this is more than 5 times. There's more than 5 times more in Arbi Shiheb than in Sukri, for example. And here you have some specific pears which are meant for pear wines, which are very rich in procyanidins. And the second thing is, beyond this variability, that the flesh and the skin cannot be predicted from one another. You can have large concentrations in the skin and small in the flesh, and vice versa. So it's always something which you have to take into account when you're talking about polyphenol content of the fruits, this variability and the differences between the different tissues. And this variability, this marked difference, is something which I've shown for pear. You find the same thing, for example, for apple. This is an example comparing common table apples and cider apples, with always a higher concentration in cider apples, which is important for the mouthfeel and the sensory aspects of the cider. You have the same thing here in apricots, in strawberries, in plums. So this is a very typical thing about polyphenols, that they will vary a lot because they are secondary metabolites between the different varieties of the same species. And the conclusion of that is that if somebody tells you this or this fruit is very rich in polyphenol, then you have to think twice. Is it really the species, or is it that I'm looking at a variety which is very rich, and if I look at another, maybe I will be disappointed? And so that's an important fact to keep in mind. The second thing is, if we look at the variability not in a static way, but in a kinetic way, so what happens to the polyphenols during maturity and growth along the growth stage of the fruit and storage, but also depending on which growth conditions are applied. So this is an example of the polyphenol accumulation in apple. And I have presented it not as concentration, but in milligram per fruit. What we see is that these are two varieties of apple, Kermeria and Avrol, and this is the polyphenol concentration in milligram per fruit, the total polyphenol. And what we see is that there is for tannins, there is an accumulation which occurs very early during fruit growth. This is 30 to 60 days after full flowering. And then the concentration per fruit doesn't change significantly during fruit growth. But because this is during fruit growth, there is actually a marked decrease of the concentration, which is due to dilution with sugar accumulation during in the fruit. And this is the same thing for the hydroxycinnamic acids. And as you see, again, the contents per fruit are quite stable. And there is a parallel of a decrease in concentration as the fruit grows. And this is something which is observed more frequently, that polyphenol are accumulated early during fruit growth. If we're talking about polyphenol from a fruit tree, there will be an impact of some growing interventions. Here, that's again an example in apple of the impact of fruit thinning and maturation. Fruit thinning is applied on apple trees to have less apples per tree, and that this apple be larger. And here you have some apples which are not thinned. They are thinned to half the original amount of apples, and they're to a quarter, only 25% of the original number of apples which are allowed to grow. And what we see is that when the apples are thinned, they are larger and they accumulate more polyphenols. Again, this is in amount per fruit, in milligram per fruit. There's also some consequences on the other quality characteristics here, like dry matter concentration. and in the final ripe fruit. We have also compared in the same system what happened in milligram per fruit, not only as a function of the thinning, but also as a function of the harvest period. So this is all close to the normal commercial harvest, and you have 2 weeks before to 1 week after, and P3 would be the normal commercial harvest for these fruits. What we see is that the not thinned fruits, which are small, have less flavanol, and the concentration or the contents of flavanol per fruit increase with the thinning, and we see some limited changes during maturation, because this is maturation. And at the same time, if we look at the concentrations, we see again a slight effect of dilution due to fruit growth over the course of the maturation. So this is the conclusion of this, is that we can see this effect of fruit growth and the modifications which are not so important close to harvest. This is a quite stable composition at that point. And we have also looked at what happened, in fact, if you're looking at the whole cultivation system. This is an experiment where we compared 3 varieties which were grown in the same orchards by the same person, under the same irrigation, the same level of fertilization, the same modes of thinning, and the difference between the 3 systems were that they were grown under conventional growing system. They were grown under low input growing system, where you limit as much as possible all chemical interventions, all interventions for control of pests, pesticides as more generally. And the third is organic. So when only organic methods are applied for the protection of the fruit. And the 3 varieties, Ariane, Melrose, Smoothie, which is a kind of golden, 3 years, 2011, 2012, and 2013. And this is the sum of the polyphenol in milligram per kilo fresh weight. And what we see is that the differences are mostly due to the variety. And we did on that experiment quite a deep statistics dive in, and there are few significant differences due to the growing system. The first difference is due to the variety. Then we can see a significant impact of year. But we see the impact of growing system only on a few of the polyphenols, and not every year the same difference. So what we see is that the impact of the cultivation system per se, in terms of how the crop protection is carried out, is quite limited. Probably because the other factors will be more prevalent. Variety, pedoclimatic conditions, and also other interventions like thinning or irrigation. So now that we've seen all this variability which exists in the fresh fruit, what happens if we now look at the juice and the cider? I will first give you a few words in description about French cider. French cider is a type of hard cider which is only partly fermented, which means that in the final drink there is alcohol, but there are still sugars also. It's slightly fizzy, and it's yellow to orange. It has a high polyphenol content, because we are using those specific special apple cultivars, which have a very high content of polyphenols and notably a very high content of tannins. In the final drink, there is a slight bitterness and astringency, and it has strong antioxidant properties equivalent to red wines. The production of cider is slightly different to what happens with some other fermented beverages like wine, because in fact, in cider fermentation, we attempt to have a slow fermentation and not complete fermentation. So there is, for example, no addition of ammonia, of nitrogen to reach total fermentation. So the apples are ground and they are pressed for extraction, and the juice is clarified. Then there will be a fermentation, and to stop fermentation or slow fermentation, because it's actually slowing fermentation, the yeast biomass is eliminated, and then a slow fermentation goes on until bottling, and all of this is carried at temperatures around 10 to 15 degrees. So it's slow, it's colder, slower than wine, and not complete. That's the main typical points of a French cider fermentation. When we look at polyphenols, we see that major modifications occur during extraction of the juice. You have here an example with apples. There's 5 varieties of apple: Douce Coëtligné, Douce Moine, Guilvic, Kermeria, Petit Jaune. They all have quite high concentrations of tannins, of procyanidins, and some of them have no monomeric catechins. They can have hydroxycinnamic acid in varying amounts, and also some dihydrochalcones and minor amounts of flavonols. Please pay attention to Guilvic, Gu here, which has no monomeric catechins. It has very high degree of polymerization, procyanidins, usually between 15 and 30. And what happens when these apples are pressed? We end up with a juice, which has a very different composition. The procyanidins have been lost, or at least are present in much lower amounts. We also have less hydroxycinnamic acids, less dihydrochalcones, and almost no flavonol. And in apple fermentation, there is not a lot that happens between the juice and the cider. And as you can see here for Guilvic, almost all the procyanidins have disappeared. So that was a very puzzling observation. What happened to all these high molecular weight tannins? So what happened to the polyphenols? When we look at those polyphenols, we see that a lot of them are actually retained in the pomace, in the solids, and a lower amount goes into the juice, the must. And we also observe that the degree of polymerization of the procyanidins changes a lot. It is much higher in the pomace than in the must in the juice. And actually, when we look at it, we see that there are 2 phenomena which occur. One is that the polyphenols, which are originally present in the vacuole, are retained by the cell walls during pressing. And in apple juice, there is no or almost no incubation of the ground fruit on its cell walls. It's very fast. It's pressed quite quickly after grinding. So there is no occasion for release. And the second thing which happens is oxidation by the polyphenol oxidases, which are present in the chloroplasts. So the first part is that a major phenomenon is that the tannins, the loss of the tannins, can be linked to their retention by the cell walls. And this was demonstrated in a model system where we had isolated cell walls and isolated procyanidins. This adsorption can be modeled as a function of concentration of the polyphenols here, the total concentration of procyanidins. And here we measure by difference the amount which is bound on the cell wall. There is a major effect of the degree of polymerization. You have here 4 examples. Degree of polymerization of 2, it's retained, but not a lot. Most of it will go to the must. Degree of polymerization of 4, it's already quite more retained. And degree of polymerization of 73, in fact, most of the polyphenols, most of the tannins, are retained on the cell walls. This can be done in the model system, isolated polyphenol, isolated cell walls, but it also occurs during pressing. And this is an example where this adsorption can be modulated by temperature of pressing. It's the same apples here which we find, which are Guilvic, very high degree of polymerization, Kermerien and Douce Moins, which are 2 apples which have a high concentration of procyanidins, but they are not so large. And what we have looked at is what happens when you grind and press the apples at temperatures between 4 degrees and 25 degrees Celsius, which is quite realistic for processing to cider. Mostly it will occur between 11 and 18. That's more like the common temperature. And we have measured how much of the procyanidins were found in the juice. For Guilvic, it's less than 5%. So we find only less than 5% of the polyphenols which were in the apple in the juice. For Kermerien or Douce Moins, it's about a third, about 30%. And what we also see is that the degree of polymerization increase with the increase in temperature. So, and in this case, this is pressing in anaerobic conditions. We have avoided as much as possible oxidation to be able to look mostly at what happens due to adsorption, because there is also oxidation occurring at the same time. So in these systems, we also see that there is a modulation of the extraction efficiency due to maturity. This is an example with pear, ripe pear here below and overripe pear above. There are some differences in composition between overripe and ripe pear in concentrations, here expressed in milligram per kilo fresh weight. And if we look at the juices, again, obtained just after pressing, we see that systematically there are lower amounts in the juice of the overripe pears than in the juice of the ripe pears. And this, whether the concentrations in the fruit itself were higher or lower in the ripe or in the overripe. So this extraction is decreased, and there's probably some link with either with the evolution of the cell walls or with the aging the cell senescence, which means that they will be more easily lost from the vacuole. There is less protection. So now what happens if we allow oxidation to take place? This is oxidation between grinding and pressing. So either we go from no oxygen, this is what we did in the beginning. Normal oxidation, we don't try to avoid oxygen or air, but we do nothing special, it's pressed quite rapidly. Or we leave it longer to oxidize. Longer is about 1 hour, or up to 4 hours with also tumbling, so it really gets oxidized. And what we see in these conditions is that the native polyphenols are totally lost in the juice with a level of oxidation. This is true for procyanidins here in red and the other flavanol, the monomers. This is true also for the dihydrochalcones, but the slope is less steep. They resist more until the very high levels of oxidation. And it is true also for the hydroxycinnamic acids. Again, we only lose them totally at the very highest level of oxidation, though there is a decrease with oxidation. And we also noticed that paracoumaroyl quinic acid, which is the orange cycle with a white middle, is more resistant to oxidation than the other polyphenols. This is important because oxidation, this elimination of the native polyphenols, also modifies the color. Actually, we go through a higher color and then it decreases again. And it also modifies the in-mouth perception. This is the same juices, This is the same juices obtained at 4 different levels of oxidation for the 3 apples varieties I have discussed before. Douce-Moins, Guivic have very high molecular weight and almost only tannins, and Kermerien. The difference between Douce-Moins and Kermerien is mostly that Kermerien will have more oxidation, more hydroxycinnamic acids. Depending on the level of oxidation, at the start we have juices which are quite astringent and also bitter. And with oxidation, we see a fast decrease of the bitterness and also a decrease of the astringency when the fruit is more oxidized before pressing. This is true mostly for Kermerien and Douce-Moins. For Guivic is much less sensitive to oxidation. It doesn't have as much hydroxycinnamic acids, which are the motors actually of the oxidation here, and it has its tannins inhibit polyphenol oxidase. So what we see here is that bitterness and astringency both decrease when the pulp is oxidized before pressing. So it's also a way to modulate this bitterness and astringency. We have an apple, Kermerien, which is more bitter and astringent. We have Guivic, which is not so astringent, but the astringency persists. And we see this very strong interaction between variety and oxidation because the behaviors are quite different for the 3 apples. I started to talk to caffeoylquinic acid being the motor of this oxidation. Actually, what happens is that the polyphenol oxidase oxidizes caffeoylquinic acid to form quinones. And what happens later on is that those quinones of caffeoylquinic will react with the other polyphenols, with themselves, and they will form adducts, covalent adducts. Prior to clarification, and you have different types of adducts which are formed between 2 caffeoylquinic acid, with caffeoylquinic acid and monomeric flavanol, or with procyanidins. And this is very typical. We don't see so much polymerization, mostly just divalent dimers and their evolution. And just a few words about what happens when you heat instead of letting the polyphenol oxidase have its play. When we look at what happens during thermal processing and refining, this is an example with puree. We see here that the polyphenol composition in the puree is very close to that of the flesh of the fruit. If you prepare a puree, you will heat the fruit and you will sieve it to eliminate all the hard bits, including the skin. You have here 2 apples, golden and granny. This is the composition of the skin, a very high, a much higher polyphenol concentration than in the flesh all the time. And what happens is that, as you can see, the composition in the puree in milligram per kilo fresh weight is almost the same as in the flesh. But we do notice that there is some oxidation in the early stages, and that also explains the change in color. Here we have the presence of fluorescein oxidation product with a very typical signal in the HPLC, and one of these products is yellow, and it gives a typical yellow color. So what we see here, that there is time for some, but not a lot of reaction, and the composition is that mostly of the flesh. And this also happens when you are looking not at puree, where everything is ground, but when canned fruits. You have here the fresh fruit, that's apricot, canned apricot, and apricot is very difficult to peel, so here you have everything. Canning is a much more intense heat process, so here you have a loss between the fresh fruit here and the canned fruit. And you see that there are some polyphenols which have been lost to the juice, and the evolution goes on with always mostly a retention of the polyphenols, and especially the procyanidins, which are in red in the fruit. And it's true for different varieties of apricot. So there is some loss upon cooking, and there is some leaching to the syrup, and except of the procyanidins, and there is an ongoing degradation during storage. So my conclusions on this topic. One is that the statement that a species is richer in polyphenol or very rich in polyphenols, I think it is misleading, because there is so much variation within the species for the fruits from different varieties. And if we look at this, how the variability can be seen, we have qualitative variability. The types of polyphenol which are present depend on the species mostly, and then there is variation per variety, but you have a very strong determination by the species of exactly which polyphenol will be present. And there is also a quantitative variability. There's a major factor is genetics, variety, then the pedoclimatic condition, then the growth system. The effect of the process will be process dependent. For example, we see that there is much more differences in polyphenol composition between a fruit and a juice than between a fruit and a puree or a canned fruit. And I looked at 2 main mechanisms, which are retention of the polyphenol in the fruit or on the pomace of the fruit due to adsorption on the cell wall on the insoluble parts versus extraction. And the second mechanism is a chemical reactivity, mostly oxidation for the short times which are realistic for apple and its apple juice and cider. And another conclusion is that to be able to make valid conclusions on the fate of polyphenols during processing, it's important to have a comprehensive analysis of polyphenols. The stories which I've just told you would be very much different if I had only looked at hydroxycinamic acid or flavonols. So it's important to take the care and pain to include the tannins in the analysis. And this is the end of the story. And I would like to thank you all for your attention. And I would like to thank all my colleagues with whom I have done this work. This is a very short summary of work which was carried out over 20 years, from 1998 to 2018, and here all the people who have been involved. Thank you for your attention.
Thank you very much, Catherine. That was a great presentation. I really enjoy it, and that's probably because I'm dealing with the same kind of issues or problematics, same questions. So it was really interesting. Thank you for that.
Thank you.
So I would like to mention that if you have any question, I just sent a message in the chat box. So please type any question you have for Dr. Renard in the chat box, and we will answer those questions at the end of the presentation. So now I would like to introduce Dr. Elsa Brandão, who I met, I think, the first time at the ICP conference I attended, I believe, a few years ago. And so Elsa Brandão is a researcher at LAQV Requimpt, Faculty of Science of Porto. In 2018, she obtained her PhD in Sustainable Chemistry by University of Bordeaux, University of Aveiro, and University Nova de Lisboa. In 2019, she won a Junior Individual Research Position, CEEC, in a highly competitive national call. Her research is focused on the understanding of taste properties, astringency and bitterness, of polyphenol-rich products, the underlying molecular mechanism, and their modulation. She has been developing economical, innovative, and sustainable approaches to modulate these unpleasant taste properties by using natural compounds, such as polysaccharides, as well as other compounds. Elsa published 27 articles in peer-reviewed journals, 1 book chapter, and she frequently participates in international scientific conferences and workshops. She has been tutoring and supervising various graduate students and is currently the co-supervisor of a PhD student. She has been involved in several projects, Biopad, Food Nanoscience, Iberphenol, Mob Food, and Yes Yeast, and is a project member of an ERC and a European project with Biome. Elsa has also established various collaborations with national and European research groups, Supagro Montpellier, France, and with Portuguese industries and companies. So Elsa, I will make you presenter when I can. And you will be able to start sharing your screen.
So good afternoon all. Thank you all for your kind words. It's a very complete description. And today in this webinar, I will talk about the complex relationship between quality and mouthfeel properties, and specifically in the case of wine. So, wine or red wine is the oldest and the most popular beverage around the world due to their remarkable sensory properties such as aroma, flavor, mouthfeel, color, turbidity, and unpleasant odors. And as well as the complexity of the matrix itself, red wines have also complicated profiles of mouthfeel perceptions, for example, body, burning, balance, warmth, viscosity, and of course, astringency that can act individually or in combination. In fact, astringency is one of the most important parameters for wine quality, which has a strong influence on consumers' acceptability, not only toward red wine, but also toward different polyphenol-rich products. So, what is wine quality? Wine quality is a very complex definition. Renard and Marin was wine researchers in the last century who once said that wine quality is easier to detect than define, and it is true because wine quality is the result of complex interactions between several factors. For example, geological soil variables, climate, and viticulture decisions. Taking as a whole all these factors, we can call the terroir effect. But wine quality is also influenced by vinification process, for example, harvest, maceration, fermentation, and also aging in barrels. Concerning climate, it's important to refer that climate has a great impact on wine excellence because it will affect the berry temperature, influencing the fruit composition, and at the end, influencing, of course, the sensory properties of wine. So, if it is difficult to define wine quality, it's also difficult to assess this property. So, the question is, wine quality assessment is holding hands with wine certification, and this evaluation process is very important, not only to improve wine quality, but also to stratify wines and setting prices. So, wine quality is often assessed by a combination of instrumental and sensory analysis. In instrumental analysis, we have different physical chemical properties that can be determined. For example, volatile acidity, residual sugars, pH, the content of sulfur, sulfates, alcohol, among others. And we can do this by different instruments and techniques. But we also have colorimetric assays, for example, to estimate the total polyphenol content of wine. Concerning the sensory analysis, these kind of analyses are carried out by human experts, well trained to distinguish the different sensory properties. But the sensory analysis is a very subjective process because it depends of cultural habits, training, experience, and of course it will affect consumers' acceptance and preference. And this makes wine classification a difficult task. The question is, it is curious because taste is perhaps the less understood of the human senses. And this paradigm has been changing in the last years, hopefully. And the problem is that the wine quality assessment now is dependent of the combination of these analyses and is not fully understood because it is a very complex combination, instrument analysis and sensory analysis. So, what are mouthfeel properties? As wines are reported to a least complex mouthfeel properties, mouthfeel wheel was developed for red wines and later for white wines. This mouthfeel wheel was established based on the importance for wine tasters to achieve a common understanding of terms describing wine mouthfeel. So this mouthfeel wheel is a suggested list of terms describing wine taste attributes, and it was made to turn clear communication among the several wine experts. So as you can see here, astringency, the astringent mouthfeel has more than half part of this wheel. And in each astringency mouthfeel, we have different astringency subqualities. This is very important because the sensory attributes described here are a useful tool for continually improving red wine quality. Additionally, control of wine mouthfeel during production could be a valuable strategy to meet consumers' wine preference and increasing its acceptability. So we are talking about mouthfeel, but which compounds are responsible for this? Which is the importance of this? So when you have wine, the wine is mainly composed by water and ethanol, but then we have 3%, and this 3% makes the difference between all wines, and this 3% brings the magic. This 3% represents several compounds and wine constituents such as glycerol, organic acids, amino acids, sugars, but we have also polyphenols, and polyphenols are the key compounds for wine quality. And among the different families that are important for wine mouthfeel, we have anthocyanins, mouthfeel and sensory property, because anthocyanins are widely known to contribute for wine color. But anthocyanins are also responsible or also reported to be involved in tactile sensations in the mouth. And anthocyanins are also reported to be bitter, persistent, and to cause dryness in the oral cavity. Then we also have phenolic acids. It's another important family of polyphenols, and in specifically gallic acid. Gallic acid is the most abundant phenolic acid in wine, and its concentration is around 70 milligrams per liter. And then we have flavanols. Flavanols are like and condensed tannins are like the champions of mouthfeel properties in wine. We have epicatechin because it's the most abundant flavanotrozole in wine, followed by catechin. But we also can find other flavanotrozoles and condensed tannins, such as B-type porcyanidins, trimercium, and also epigallocatechin and epigalloggalate. And this is very important because these polyphenols are, in fact, responsible for the sensory properties and more specifically for mouthfeel in wine. And another thing is that the tannin content is one of the most important parameters influencing astringency, and specifically the sub-quality of dryness. Additionally, other polyphenols features, for example, the hydroxylation of the B ring, as well as the condensation of flavanols, are also important to the coarse astringency taste. On the other hand, acylation is very important to contribute to grassy astringent taste. Another thing that could be important to refer here is that, for example, it's expected that the astringency increases as the polymerization of polyphenols increases as well as acylation. The polyphenol content, either qualitatively or quantitatively, largely define the quality of wine due to their contribution to its sensory properties. So, when we talk about mouthfeel property, we need to talk about astringency, the most known mouthfeel property. And astringency is a tactile sensation, including dryness, pugness, tightening, and pulpy sensations felt in the oral cavity. Astringency increases with repeated exposures, and the rate of increase is greater when the time of between exposures is shortened. So, if high quality wines have a balanced level of astringency, it's important to maintain this balanced level. When astringency it is felt in excess, they can detract from other components, being an undesirable property. On the other hand, when astringency is low, wine may taste flat and insipid. So, a acceptable level of astringency needs a balance. Astringency is also important to contribute for body and fullness to red wine. Indeed, body is the weight of wine in the palate and is the result of the combination of the perception of combined compounds or constituents such as alcohol, sugar, tannins, acid, and glycerol. The problem is that nowadays, consumers are demanding softer astringency along with full-body wines. And this is the big challenge for winemakers to create wines with a balanced level of astringency as the time they have a full body. So, astringency can be related to different mechanisms, molecular mechanisms. And the most known mechanism to explain astringency on set relies on the interaction between salivary proteins and tannins that can form soluble complexes, and then they can bind other proteins or even other tannins or other complexes, forming bigger complexes that will precipitate, causing the disruption of saliva film and consequently the loss of lubrication. This is the most known, the most studied mechanism. But the truth is, now we know that probably astringency is not related to one mechanism, but a combination of mechanisms. And you also have the interaction with oral cells. The different polyphenols, in this case tannins, can interact with oral cells or even with other oral constituents. And the third mechanism, the less studied one, refers that tannins or other polyphenols can bind specific kind of receptors called mechanoreceptors. And now I will show some results that we have concerning wine and grapes, of course, and the correlation with astringency, mouthfeel, also astringency, the molecular mechanisms of astringency. And here, this study was performed a few years ago, and several volunteers gave repeated sips of a grape seed extract solution, in this case 5. The sips were given with 3 minutes intervals, and the last exposure, the last exposure, saliva was collecting during 16 minutes, and the saliva from different volunteers were pulled together. And the results show that some families of salivary proteins decrease its percentage in mouth because they interact. They have a higher interaction with this extract, and this is particularly evident for acidic PRPs and staterine. That after in this, we need to note that staterine was almost depleted after the exposures of grape seed extract. And in the right side, the graph, it is important to note that is the saliva collected after finishing the repeated exposures. It is possible to note that saliva was collected 5, 30, and 60 minutes after the last exposure, 1 hour after. And it is possible to observe that in the case, for example, of glycosylated PRPs, nothing happened, which means this family of salivary proteins did not recover its percentage in the mouth. So we need to pay attention to acidic PRPs and staterine. It is possible to note that for acidic PRPs, 5 minutes after the last exposure, its content is about half of the initial content. And even after 1 hour later, we don't reach the initial concentration of salivary proteins. In the case of staterine, we need 30 minutes to achieve 25% of the initial content. And similar to what was observed for acidic PRPs, we didn't reach the maximum, which means we didn't reach the initial content of salivary proteins. These results probably suggest that after the last exposure, the proteins that were stored in the salivary glands were ready to be released. But then we need more time to a new synthesis of these proteins to reach the initial amount of salivary proteins in the mouth. Another interesting study refers to the nebule red wines and their wine analysis, as well as sensory analysis and the interaction with salivary proteins. These wines were considered astringent by the trained panel. And we have 2 wines, P and M, the codes given by the industry because it was made in collaboration with wine industry. And first, the 2 wines were analyzed in terms of specific methods to characterize these wines. For example, the total polyphenol content was analyzed by Fuller. We also analyzed tannin-specific activity by turbidity, and we also analyzed the total of proanthocyanidins. It is possible to note that in all of these parameters analyzed, the wine M showed the higher results. And then we went to analyze the different interactions of these wines with salivary proteins, and we used 3 different rations: salivary proteins, wine. And the truth is, in the lowest ratio, we observed that most families of salivary proteins decrease their concentration, which means that they have a higher interaction with wine. We have here the numbers represent the bottle replicates, and we observe that Acid PRPs, statin and PB peptide, as well as cystatin, interact with these wines. But when we increased the ratio, it was observed that not only these families continued to interact at in the higher rate of interaction, but also we observed that PPRPs and PPRPs also interact. And these altogether, these results show or suggest that because even the interaction was higher for wine P than for wine M than wine P. So altogether, these results suggest that wine M is more astringent than wine P. Another interesting study refers to the needing of food industry, and more specifically wine industry, to have a wine biosensor. This biosensor is an electrochemical biosensor that is based on electrostatic affinity between salivary proteins and polyphenols, and the affinity was tracked by electrical changes in a typical behavior of a standard iron redox proof that was measured by cyclic voltammetry and also electrochemical intense spectroscopy. And this biosensor is composed by a golden part, the sensing area, then with a slight modification to with cysteamine. And then we have the absorption of salivary proteins, in this case PRP and the alpha amylase. And after this, we have the binding of PTG. PTG was used because it was a polyphenol model for astringency. And we need to validate this not only for pure compounds, such as PGT, because we know that it is astringency, and the biosensor gave the signal like this, but we also need to validate these results in wine. So we have 3 wines: red wine, white wine, and rosé wine. And it was asked to the trained panel to classify the different wines. And it is possible to note that red wine was classified as high astringent, must wine low astringent, and rosé wine no astringency was detected. And then we will check and validate the results by the biosensor. And we observe that red wine 1 and 2, because the difference of red wines 1 and 2 is referred to the presence of yeast protein extract, because wine 2 was treated with this yeast extract. It is possible to note that red wines, which were classified as high astringent, have the higher signal of the biosensor. And this signal was referred to the PGG equivalent. We also can see that rosé wine or must wine have lower signals, and at the same time it was also tested the effect of side compounds also present in wine, for example glucose, fructose, glycerol, and ethanol, on this biosensor. And as you can see, the signal was practically inexistent. So we are talking about mouthfeel, wine mouthfeel, but in the end we also talking about a balance level. But how we can get it? How we can soft astringency? It's a very important question. For example, we can use polysaccharides. So in this study, different fractions of polysaccharides that were obtained from grape skins were added to a wine without treatment. This wine was a green wine from north of Portugal. And we also use not only polysaccharide fractions obtained from grape skins, but also commercial polysaccharides such as pectin and arabica. And the wines were supplemented with these polysaccharides and were kept in storage for 9 months. During these 9 months, at every 3 months, we analyzed several parameters and we performed different assays. So it is possible to note or to observe that in this case, we have the wine treated with carbonate polysaccharides, and we also have the wine treated with imidazole polysaccharides. And the bifola, this is the a quick method to estimate the total polyphenol content. It's possible to note in the presence of wine treated, polyphenol content decreased. Then we need to note or to study what happens, for example, by tannin-specific activity, by turbidity. And BSA is usually or commonly a standard protein to study turbidity. But we also want to study this tannin-specific activity toward salivary proteins because it's our core of astringency, and also mucin. Mucin is a glycoprotein very important to mouth lubrication. And it is possible to note that only turbidity was higher toward salivary proteins, and in this case, in the control wine and even the treated wines. But it's important to note that the wine treated with carbonate, we observed a decrease of turbidity in comparison with the control wine and also with the wine treated with imidazole. And then we can move to the results obtained by interaction with salivary proteins by HPLC. And it's possible to note that different families of salivary proteins were affected or were interacted with wine constituents. These families are acidic PRPs, staturin, PB, and cystatins. And it's important to observe that in the case of wine treated with the carbonate polysaccharides, soluble polysaccharides, it is possible to observe that this, which means this wine caused a lower interaction with salivary proteins in comparison with the control wine. This was observed for these four families of salivary proteins: acidic PRPs, staturin peptide, and cystatins. This is very important because are in agreement with the previous results. Also, the wine treated with imidazole also decreases the total polyphenol content, was not able to decrease in the same extension in the number of salivary proteins as the wine treated with carbonate. The same case. For example, in the case of the wine treated with imidazole, we can observe a decrease of interaction only for acidic PRPs and staturin, but not for the other families of salivary proteins. Altogether, these results can suggest that the decrease of turbidity, the decrease of interaction with salivary proteins, we can say, or it's possible, related to a decrease of astringency. So polysaccharides added to wine maybe could be a valuable tool to balance the level of astringency. As concluding remarks, it is important to say that wine sector is undergoing several transformations as a result of climate changes, as well as increasing consumers' expectations, and the market is a highly competitive market. So high-quality wines is the production of high-quality wines is a big challenge for winemakers, because they need to increase the production of compounds with positive contribution to wines, and they need to decrease or limit the production of compounds that affect negatively wine quality, for example, of flavors. But the question is there a perfect wine? Probably not. What is perfect for me is not perfect for you, because the relationship between the properties that are related to wine quality is a very complex phenomenon. Wine quality depends of interaction of several properties, and these properties are dependent of intrinsic and extrinsic factors. And when we are talking about sensory properties, we need to mention mouthfeel properties. And we are talking about mouthfeel properties, we need to talk about astringency. And astringency is probably the most difficult phenomenon for winemakers to overcome and to create strategies and to develop tools to modulate it. Because astringency is related to the polyphenol content, in specific as we refer tannins, we need to take into account the mechanisms, and we need to reach this balanced level. But what is considered a balanced level and how we get it? But you also need to be to pay attention to the individual subjectivity and the different compounds that can be used to modulate it. So altogether, these parameters and properties influence the final profile of compounds present in wine that will affect the overall wine quality. And this profile, the compounds present are mainly, as we referred, polyphenols. So the future is to validate and to develop and validate strategies to overcome these problems and these properties. So to finish, I'd like to thank all the institutions for the financial support, as well as my colleagues, and all of you for your attention. Thank you very much.
Thank you very much, Elsa. It was a great presentation about red wine quality, right? But very interesting. So now, everyone, if you have any questions, please use the chat box to type your question, and that's the time to answer them. So I already have a few questions. Maybe some of them will be available to you panelists, maybe not. So I'm gonna read some of them. So the first question can be for both of you. It's about the method to tannin analysis. What would you recommend to measure how much tannin you have in a fruit or in a juice or in a wine or other beverage? Catherine, you want to start?
I would recommend one of the solvolysis-based methods. Whether it is using thiolysis, whether it is using I don't remember the name. It's coming back. But I would recommend using one of the solvolysis methods, where you actually cleave the tannins into monomers, and you can measure the monomers, because you have at the same time a quantitative information and also an information about the composition. However, it doesn't always work, because if you have type R procyanidins, then they are not cleaved and the picture becomes more complex. Alternatively, the classical HCl method will also work and give good quantitative results, but it's purely quantitative.
Mm-hmm. Yeah. Do you have any other ideas, Elsa, about tannin? What technique do you use?
We usually use HPLC because we have a well-established method to analyze tannins by HPLC. So we can identify, for example, the type of condensed tannins, as well as we can quantify the amount in wine. We can do estimation and estimation. Yeah.
Yeah.
But of course, we need sometimes to do some treatments to analyze this kind of complex matrix. Yeah.
Mm-hmm. Yeah. Thank you.
Solvolysis means HPLC afterwards. Sorry.
Mm-hmm. Yeah. So we have more comment from Cynthia. I believe that the organic cultivation, and that's for your presentation, Catherine. I believe that the organic cultivation system could contribute to an increase in polyphenols, but the data presented by Dr. Catherine Renard demonstrate that there is no increase in their synthesis in this production system. I found this data quite interesting. Congratulations on the lecture. But yeah, I think that was surprising to see that the conventional versus organic production system didn't impact anything in terms of concentration of polyphenols. Is it surprising to you, or is it what you expected to have?
How can I say that? It's both surprising and not really. because when we started looking at this, and that's quite a long time ago, we also expected to have higher concentrations in organic, because it seemed to be the generally accepted fact. When we have looked, whenever we have measured microconstituents in conventional or organic fruits, we have almost never found significant differences. But it's also a matter of the way you look, of the way you carry out your work. Because if you go to the supermarket and you go to the organic ale or you go to the conventional ale, you will have different varieties. And this will impact. But it is a matter of which variety is more appropriate for organic cultivation and which variety is more appropriate or only appropriate for conventional cultivation methods. So what you see is, if you take as a system the whole thing, including the variety, then you will have differences because you do not have the same variety. If you have also cultivation which is carried out by different people in different places, then you will also probably have differences which are due to other factors than just the protection system, because they will have different irrigations, maybe different levels of fertilization, but none of that is part of the definition of organic versus conventional. So if you just compare an organic system versus a conventional system, and the only difference is what corresponds to the definition, which is the mode of protection and the fact that you're not using synthetic fertilizers, but it doesn't say how much, then you have no difference. The differences are due to other factors.
Mm-hmm. And probably you would have a difference if you apply a specific nutrient initially at the beginning of the growth of the fruit. Like if you apply a nutrient that is rich in nitrogen, maybe that would help to change a little bit the biosynthesis of polyphenols.
Yeah, if you have higher nitrogen, then you will have higher synthesis of. Yeah, it will change the biosynthesis, and it's also something which is very much discussed about people who are working on plant physiology. Because if you want to synthesize secondary metabolites, it takes a lot of energy, a lot of enzymes. So are they more synthesized when the plant is under stress, or are they more synthesized when the plant has enough energy to make all this machinery, all these enzymes and everything, and divert all this energy to doing the secondary metabolites? So that's very much something which is still discussed.
Yeah, thank you. So we have a question from Magdalena, who is saying, so it was a great presentation. I have just one question. We are trying to transfer as much polyphenols to the final product as possible. And I'm not sure what product she's talking about, but is there any limit of polyphenol that we can consume per day? So I think that's more related to bioavailability question.
Yes, maybe. We can know the recommended polyphenol intake, daily intake, but I don't know how much can be dangerous for health.
It's really a difficult question because polyphenols are not vitamins, so there are no actual recommendations. Polyphenols have, and also because if you're eating more polyphenol, you will have also an impact on, depending on what you eat them with, you will have an impact on digestibility of proteins or the foods. You will limit the oxidation of lipids, but it will also depend on whether you have a system which we have a plethora of food. Then the fact that you limit the protein digestion is not a problem. Or if you have a system where there is not enough food and then the anti-nutrient property comes to the fore. So no, there is no recommendation of how much polyphenol is too much polyphenol at this stage, I think. And I don't think it's really a matter of bioavailability, because many polyphenols have very low bioavailability. What becomes really bioavailable is a colonic degradation metabolites.
Mm-hmm. Yeah, so I don't think we have an answer for you, Magdalena, at this time, but soon, maybe. So then a question for you, Elsa. Do you know any information about the importance of molecular association between wine polyphenols and wine polysaccharides to smooth astringency?
I didn't understand the beginning. So the relationship between wine polysaccharides and wine polyphenols?
So how molecular interactions or association are happening between polyphenols and polysaccharides in wine, and how that really changed the astringency?
Yeah. Suppose that before interact with salivary proteins, polyphenols and polysaccharides interact in food matrices, in this case wine matrices. And these kind of interactions, of course, will affect the interaction with salivary proteins. Indeed, in our assays, usually we use the first to interact polysaccharides with polyphenols to mimic what really happens in the food and beverages matrices, and then we have this solution to our assays with salivary proteins or oral constituents. And of course, we know that polysaccharides and polyphenols interact before, and the kind of interactions will affect astringency. Indeed, we know that depending on the polysaccharide mechanisms, even polysaccharides are able not only to interact with polyphenols and even to precipitate it. Yes, we already saw this in some assays, and of course, in this way, they will affect the mechanisms, the underlying mechanisms of astringency.
Mm-hmm. And I was thinking, when you did present about your study of different type of polysaccharides added to the wines, I was thinking, why don't you try to add just soluble proteins to remove a little bit of tannin to soften the astringency?
Yes, usually the industries have the fining agents to clear the wine, but we want not to precipitate polyphenols, because polyphenols, of course, we know that they have potential health benefits. So we not only want to decrease of the polyphenols, and of course we are losing some potential health benefits, but we want to join all the benefits together. That is, not decrease so much the content of polyphenols along with the good taste. So the use of other compounds, we are studying this in preliminary results. And we are trying to use other compounds, not only polysaccharides, but our main objective is not to decrease totally or a great part of polyphenols present in wine.
Yeah, good, thank you. So now another question for Catherine. Do you think that the conclusion drawn for the impact of processing on polyphenol contents of fruits could be the same for the vegetables? Example, you said that polyphenols present in the puree are very close to those of the flesh of the fruits. Could we expect the same for vegetables used as a puree? I thank you. Marie-Laure Gontier from the University of La Réunion in France.
Yes, I think it would be very much the same for vegetables. Don't forget that many vegetables are actually fruits. Mm-hmm. But it's really a matter of dealing with the question of the skin versus the flesh. And also, if you're talking about vegetables, very often they are more heat processed than the fruits because they have a higher pH. And when I say that, this is again a generalization, because it's not true, for example, of tomato. Tomato is acidic, and there's not much happening to its polyphenols during processing either. But yes, generally speaking, you read a lot that polyphenols are very fragile molecules. They are not. What they are very vulnerable to is oxidation with the polyphenol oxidase. If you do a heat treatment, then you will inactivate the polyphenol oxidase, and you will find that the polyphenols are quite resistant, especially if you're measuring them using a colorimetric method. And that's what will be important afterwards.
Yeah. And on that note about polyphenol oxidase, I have a question. Do you know if the activity of polyphenol oxidase would be different depending on the variety of fruits or depending on the skins versus flesh? And how can we determine how much polyphenol oxidase is in those different tissues and varieties?
Yeah, we have measured that sometime. There is a difference. There are differences between varieties. How to measure it? The difficulty is that you have to separate the enzyme from its substrate, so you cannot just grind or juice. You have to really extract. For example, in an acetone powder, you can collect the polyphenol oxidase. Or but you really have to maybe not isolate the polyphenol oxidase, but you really have to separate it from its substrate and to be able to do really an independent determination, or maybe using oxygen loss, but adding substrate, not just the substrate from the juice, because that will interfere.
Yeah, good. So we also have a question from Shamali, who is asking, could you kindly tell us the extraction solvent you used? And I'm not sure to who she's referring or he's referring. So if you are still with us, can you please specify that question? So another question, does the fermentation process impact the antioxidant potential? So when making cider, when making wine, is fermentation, so alcohol production, impacting antioxidant potential?
Elsa, I don't know if you have a word to say on that.
You're on mute, I think. No.
Okay. No, I'm not. So the question is, does the fermentation process affect antioxidant?
Yeah.
It's a difficult question because actually the microorganisms themselves may generate enzymes, molecules which will act on the antioxidant potential. They will change, that's for sure, the redox potential of the drink. They may retain some polyphenols on the lees, for example. But it's difficult to have a simple answer to this question, I think.
It depends the material and the kind of microorganisms.
Yeah. Another question from Lucia. In my knowledge, the exposure to sun is the main condition for the growth of polyphenols in plants. Therefore, the species from Tunisia were the richest in all types of phenolics. So yeah, that's mainly a comment. That's not really a question, but sun exposure is gonna have an impact on the growth and the concentration of polyphenols you will have in the fruit. Is there anything you want to add, Catherine?
Yes, the sun exposure does have an impact, mostly on the skin, but in the experiments I showed, you had Tunisian pears and you had cider pears, and the cider pears are grown in Brittany and Normandy, which are neither hot nor very sunny. And they have as much polyphenols as the pears from Tunisia. So maybe there has been something during the selection for the growth, but it's not a direct impact of growing in Tunisia or growing in Normandy.
And the soil can change something, and everything else can have an impact, right? Yeah. Another question. Oh, we are running out of time, but last question, and we will answer to other questions if you need by email. So Elsa mentioned that polysaccharides added can be used to control the astringency level in wine. The question is it possible for wine producer to add polysaccharides to wine according to regulation for wines? So that's really, I think, the question about legal addition, or how much can you really add?
It's a good question, in fact. At this moment, we are only looking for the results at molecular level. Then, of course, we need to see the legal issues, because probably there is a legal important to take into account. But the question is, we are using, or we are looking to use compounds that will be safe for human consumption, like they are crash. We will not use, of course, compounds that will not be able to be consumed by humans. Then the concentration, we need to, of course, it's a very difficult question, because we need to use a concentration that will have effect at the same time that you need to, of course, to cover this kind of issues. So it's the same question. It's a balanced level of both these issues.
Yeah, that's challenging question. All right, so now, we are really running out of time. So I hope you can see my screen. All right. So I just want to thank you all for attending that third webinar, and a big thank you to our two fantastic speakers today who did an amazing job just to explain the role of polyphenols in food and beverage quality. Also a big thank you to Dr. Salminen, who has been taking care of the communication of this webinar series and of the Groupe Polyphénols, and has been recording the webinar to share on the Groupe Polyphénols website and on YouTube. So if you want to go back and listen to those nice presentations today, feel free to go on the website and on YouTube, Instagram, Twitter, all those different tools, social media, where you can have access to that. Also, I would like to thank you, Dr. Baron, who is the president of the Groupe Polyphenols, helping and managing this webinar series. So just a few more notes before closing the webinar. You will receive an email. I think you already received an email, but for a survey to complete in order to know how we can improve the webinar series for other future webinars. And finally, don't forget to register and to come to the 31st International Conference on Polyphenols. That's going to happen from July 3rd to 6th this year in Nantes, in France. So please register, and I'm looking forward to seeing you all this summer. So thank you very much again, and have a good day, everyone.